CRISPR for immune evasion of cancer cells. Cancer is the uncontrolled rapid growth or lack of cell death of neoplastic cells, or a mix of both. This abnormal cellular growth produces lumps in the tissue called tumors. If the tumor is malignant, it has the potential to expand to nearby structures and destroy them, or can spread via blood and lymph to tissues at distant sites. This sets in motion a progressive deterioration of the whole organism. But who gives cells the green light to begin this insane reproduction? A gene is a section of DNA that gives instructions to create a specific protein or parts of a protein that will do something in the body, or it produces non-coding DNA. There are tiny mutations to some genes that lead to cancer. Many of these genes are called oncogenes because they are highly prone to developing cancer-causing mutations. Sometimes our DNA becomes damaged, which can also lead to these mutations. At the end of the day, cancer is completely dependent on genes, and genetics tends to be hard to defeat. But what if we told you that there is something very special that promises to end all this one day, and it's all thanks to bacteria?
Huh?
Special viruses called bacteriophages are among the greatest enemies of bacteria. To protect themselves, bacteria have innate and adaptive immune protection. CRISPR stands for clustered regularly interspaced short palindromic repeats. It is part of the adaptive immune system in bacteria. The CRISPR portion of the bacterial DNA holds a set of viral DNA mugshots of previous viral infections, small sequences of DNA that match important genes of a previous viral infection. When a virus enters the bacterium that fits the look of the mugshot, Cas9, whose gene is located near the CRISPR sequences, acts as a DNA scissor that cuts DNA to destroy the viral DNA. This gave Jennifer Doudna and Emmanuelle Charpentier the Nobel Prize of Chemistry in 2011. The discovery of how to modify this bacterial adaptive immune system has helped to develop many new tools for medicine and agriculture. One way we employ the CRISPR-Cas system is to edit DNA to repair genes. Another is to turn genes on or off. For this reason, CRISPR-Cas9 has several potential applications in oncology. First, we can develop cancer models to identify targetable genes, investigate resistance mechanisms, and understand how cancer cells modulate the effectiveness of immunotherapy. Second, CRISPR-Cas may be used to modify immune cells such that they detect altered proteins particular to a person's cancer. These cells are then safely released into the body to locate and destroy their intended target. Acute Lymphoblastic Leukemia, a type of blood cancer, is a good example that we can use to illustrate this process. ALL is the most common type of cancer in children. Immature lymphocytes are produced like crazy in the bone marrow, but they never get to mature. They just accumulate there, precluding the development of red blood cells and other important white blood cells. There is a long list of genes with specific mutations that are proven to lead to childhood ALL. Using CRISPR, we have the possibility to cut and remove a defective gene fragment and replace it with the needed gene. This way, the production of lymphoblasts would stabilize and the child's blood cell production would return to equilibrium, making him healthy and happy again. This was brought to you by the Educated Parasites. Thank you and have a nice day.